This method allows the study of proton transport activity of respiratory membrane enzymes like cytochrome B03 in the natural lipid bilayer environment.
The main advantages of this single enzyme technique is the possibility to start and stop enzymatic reaction at any moment using electrochemistry.
Using a glass syringe, transfer 200 microliters of chloroform stock of lipid polar extract from Escherichia coli into glass vials to make 5 mg aliquots. Add 50 microliters of 1 mg per milliliter ubiquinone 10 to the lipids to make the final ratio of 1 to 100 ubiquinone 10 to lipids. To the mixture, add 20 microliters of 1 mg per milliliter long wavelength fluorescent dye-labeled lipid, abbreviated FDLL. Homogenize the chloroform solution by short vortexing, and evaporate most of the chloroform under a gentle nitrogen or argon flow. Remove the chloroform traces entirely by further evaporation under vacuum for at least 1 hour. Add 312.5 microliters of 40 millimolar MOPS pH 7.4 buffer to 1 aliquot of lipids ubiquinone 10 FDLL dry mix. Mix the vortex till the lipid film is fully resuspended, followed by 2 minutes of treatment in an ultrasonic bath. Next, add 125 microliters of 25 millimolar HPTS, a pH-sensitive fluorescent dye,